My name is Aaron Korn, and today I will be presenting my experiment on the effect of HTT polyglutamine repeats on locomotor ability in Drosophila melanogaster. In this experiment, I used a negative geotaxis assay to test the effect of expressing mutated Huntington proteins with varying polyglutamine lengths in Drosophila, and found that an increase in the number of glutamine repeats seems to result in a significant decrease in locomotor ability in Drosophila. Before I go into the details of this experiment and its results, I'll give you a short introduction to what Huntington's disease is. Huntington's disease is an inherited neurodegenerative disorder that currently has no effective treatment or cure. Its primary symptoms are chorea and changes in cognition and behavior. Huntington's in humans is caused by a mutation to exon 1 of the Huntington gene in which over 35 CAG repeats accumulate. And although Huntington's is not the only disease caused by a large number of CAG repeats, it is the most common and clinically studied. In adult Drosophila, the Huntington protein is known to be important in maintaining mobility and long-term survival rates. In order to test the effect of various HTT polyQ repeat lengths, genetically modified flies expressing either 16 or 128 glutamine repeats were bred as shown here in Figure 1. Expression of the Huntington proteins was controlled using the ELA-V Gal4 UAS system, such that the protein would only be expressed in the neurons of straight-winged female Drosophila resulting from the original crosses. These females were then isolated and split into 3 populations of 8 flies each and allowed to reach 2 weeks of age before the experiment was performed. This was done to ensure that all subjects used were expressing significant amounts of Huntington protein at the time of the experiment. Each population was then tested using a negative geotaxis assay, as shown in the diagram of Figure 2, to determine relative locomotor ability. The flies were tapped to the bottom of a glass vial and allowed to climb up the vial. A timer was started, and the time it took for 4 out of the 8 flies to climb 15 centimeters up the vial was recorded. Using the data collected from the negative geotaxis assay of the 3 populations, the average time for each genotype was calculated. This data is displayed in Figure 3 as a bar graph with the standard deviations represented by the black error bars. The Q16 flies took an average of 26 seconds with a standard deviation of 26.9 seconds, while the Q128 flies took an average of 56.3 seconds with a standard deviation of 16.8 seconds. The significantly longer response of Q128 flies suggested that an increase in the number of polyglutamine repeats in the expressed Huntington protein negatively impacted locomotor ability in Drosophila. However, the large standard deviation seen in the Q16 flies warrants repeating the experiment with larger populations. Assuming that such an experiment yields similar results, it would strongly support the conclusion that an increase in the number of polyglutamine repeats results in a decrease in locomotor ability in Drosophila. In future experiments, it may be useful to study the expression of HTT polyQ in a mammalian model organism, such as mice, as their complex neurology and behavior better model that of humans. Alternatively, it may be useful to continue studying the effects of HTT polyQ on Drosophila using a larger variety of glutamine repeat lengths. In conclusion, I would like to thank Joyce Regal, Kendrick Rubino, Ruchir Chaturvedi, and Professor Kosinski Collins for all of their help in the design, execution, and presentation of this experiment. Thank you for your time and attention.